Hello students, welcome to Ligis Academy. In this session, we will be discussing the basics of cell signaling. Have you ever thought about the process by which the cells sense their environment? Like how they know the changes happening outside the cell? Of course, they need to understand everything happening inside and outside the cells, right? For the cells to grow and divide, they should recognize some signals. For example, if there is an unnecessary increase in the level of any component, cell need to find a way to decrease it. Whatever the changes are, it may be a pressure change, chemical signals, temperature variations or whatever, the cells need to respond, right? That is where the importance of cell signaling lies. Cell signaling can be defined as the communication between cells and the response within the cells and also to the extracellular environment. So the cells receive the transmitted signals, they process them and they respond appropriately. If there is an error in processing the signal, it may cause diseases like cancer, diabetes and autoimmune disorders. In some organisms like bacteria, there is a process called quorum sensing. In this process, it involves the production and detection of a signal and they respond by gene transcription regulation. So, the cell signaling is important because it maintains and it governs various biological functions. For example, cell division, the migration, development, differentiation of cells, the protein expression, apoptosis and many more. Before moving on to the details of cell signaling, let's check out the components of cell signaling. The major two terms related to this are receptors and ligands. So what do you mean by a receptor? Receptor is a class of proteins that binds to a specific ligand and activate one or more pathways, whereas ligand is the signaling molecule itself. So what happens is that the signaling molecule or the ligand, they can communicate with the target cell either by entering into the cell or by binding to the cell surface receptors. This is entirely based on the chemical nature of the signaling molecule. Because in some cases, the signaling molecules may be small and hydrophobic, so they can pass so easily through the plasma membrane and they can diffuse into the cell. But in some other cases, the molecules may be very large and hydrophilic, so they cannot diffuse through the plasma membranes. So instead of that, what happens is that those molecules will bind to the cell surface receptors which are present on the membrane of cells, and thereby they carry out the signal transduction. So in short, there is a signal molecule and the signal molecule will bind to a receptor protein in the target cell, and the receptor activates the intracellular signaling pathways which consist of signaling molecules, or they produce secondary messengers and further they finally produce a cellular response. The responses are of two types: biochemical changes or gene expression changes. Another term related to cell signaling is signal transduction. So it can be defined as the whole process of transmission of an initial signal to the cell and by evoking a response. We have already said that the receptors are those class of proteins which bind to specific ligands. So the receptors can be classified into two types: cell surface receptors and intracellular receptors. Cell surface receptors, as the term indicates, these are receptors which are present on the cellular surface, whereas intracellular receptors are those receptors which are present within the cell. So the cell surface receptors are again classified into three types: ion channel linked receptors, G protein coupled receptors and enzyme linked receptors. Coming to ion channel linked receptors, they are of two types: ligand gated channel and voltage gated channel. In case of G protein coupled receptors, they are entirely based on a protein called as G protein. Coming to the enzyme linked receptors, they are also of three types: receptor tyrosine kinase associate receptors, serine threonine kinase receptors. All these mechanisms are detailed in coming videos. So as we said, in the case of cell surface receptors, there are some signals which are small and hydrophobic, which enables them to cross the cell membrane. But some cases, there are large and hydrophilic signal molecules that couldn't cross the plasma membrane. So they are in need of cell surface receptors. For example, vasopressin acetylcholine receptors. Coming to the structure of the cell surface receptors, they are usually embedded in the plasma membranes. Suppose this is the lipid bilayer, and these cell surface receptors will be having three domains. One at the center, that is the transmembrane domain, and another one is a domain that is the extracellular domain for binding ligand. So that is ligand binding domain, that is extracellular, and also it will have an intracellular cytosolic domain. Cytosolic domain through which it communicates to the inside of the cell. So mainly the cell surface receptors are of three types: ion channel linked receptors, which is also called as ionotropic receptors, which bind a ligand and open a channel for specific ions to pass through the membrane, for example acetylcholine. The second one is G protein coupled receptors. They activate a G protein, a membrane protein, which activates further downstream enzymes or interact with ion channels, for example CAMP pathway, IP3, DAG. And the third one is enzyme linked receptors. They are having an extracellular ligand binding domain and intracellular domains associated with enzyme, for example tyrosine kinase associated receptors. Intracellular receptors. Intracellular receptors are those receptors which are present inside the cells. They are mainly in the nucleus and cytosol of the target cells. They belong to the nuclear receptor superfamily, which consists of transcription factors, so that they regulate the transcription during signaling. It consists of three domains: N-terminal activation domain, the central DNA binding domain, and the C-terminal ligand binding domain. Their signaling molecules will be hydrophobic and small, so that they can diffuse into the cells. They can be homodimers, for example in the case of steroid hormone receptors, or heterodimers, for example retinoic acid receptor, thyroid hormone receptor, etc. Now let's look into the case of steroid hormone receptors. In the absence of a signal, the steroid receptor will be bound to heat shock proteins and will be in an inactive state. So whenever there is a signal, it will lead to the dissociation of heat shock proteins and will form steroid receptor ligand complex. So thus the ligand will cause the receptor into the active condition, and the steroid receptor ligand complex will further proceed to the signal transduction. Ligands. As we have already said, ligands are the signaling molecules. The ligands that bind to cell surface receptors are of different types. For example, neurotransmitters like norepinephrine, histamine, etc. Peptide hormones like insulin, because insulin cannot cross the membrane, growth factors, or lipophilic molecules like prostaglandins, etc. And ligands that bind to intracellular receptors will be lipid soluble, so that they can diffuse across the plasma membrane and interact with the intracellular receptors which are present in cytosol or nucleus. For example, steroids, thyroxine, retinoic acid, nitric oxide, etc. Effector proteins. Effector proteins are the final targets at which the response occurs at the end of signaling. The intracellular signaling proteins alter the activity of effector proteins and evoke cellular changes such as metabolic changes, gene expression changes, or conformational changes. These proteins belong to diverse classes of molecules such as proteins, lipids, gases, or ions, etc. Coming to the signaling types, which are mainly of two: intercellular signaling and intracellular signaling. In the case of intercellular signaling, there are different types: juxtacrine, paracrine, endocrine, and autocrine signaling. So the first one is juxtacrine signaling, which is also called as contact-dependent signaling. That is, in this case, there is a direct membrane-to-membrane contact between the cells. For example, this is cell A and this is cell B. So here the signaling will be from cell A to cell B within this contact. The contact between cell A and cell B, so the signals can be transferred from this cell to the next cell. This is called as juxtacrine signaling. The second one is paracrine signaling. In this type of signaling, a cell targets a nearby cell via local mediators that are released into the extracellular space. So this will be a short distance travel. For example, the nerve muscle signaling. Here we know that suppose this is an axon and this is the dendrite of a next cell. So this is nerve cell number 1 and this is number 2, and here we know that from this axon there will be some synaptic vesicles, and the synaptic vesicles, they release a neurotransmitter, right? So these neurotransmitters act as the local mediators, and the signaling will be carried out to the dendrite of the next neuron. Endocrine signaling involves the secretion of hormones into the bloodstream for distributions. So in this type of signaling, the signals will travel a long distance. For example, endocrine cells. And the last one is autocrine signaling. In this type of signaling, the cell that releases the signal is also the target. For example, T-cell proliferation by cytokines or tumor development. So in this case, suppose this is the target cell and there is a receptor in this target cell. Okay, so from this target cells, it produces several signals, and this signal can itself bind to the receptor present within the same cell and activate downstream processes. So this is called as autocrine signaling. Coming to intracellular signaling molecules. The signals are released to the interior of the cells itself, which are transmitted by the intracellular receptors. Being proteins, most of the intracellular molecules transmit signals by producing secondary messengers or activating other effector proteins. The major secondary messengers in intracellular signaling are cyclic AMP, diacylglycerol, inositol triphosphate, and calcium ions. Mostly, phosphorylation occurs during the intracellular signaling, which activates or deactivates further enzymes. Hope you all understood the basics of cell signaling. Stay tuned to our next videos on cell signaling. Thank you. Please like, share and subscribe Ligis Academy, your educational catalyst.